Okay, guys, here we are. We are going to go beyond Mendel. So we're going to be talking about things beyond just the simple dominant and recessive alleles. And if you mix the two genes or two alleles, only the dominant trait shows up. Okay, so we are getting into what is called non-Mendelian genetics. Okay, these are exception to Mendel's principles. Even though his principles hold true for quite a few traits, we have found out since the time of Mendel that there are some that do not. Okay, I mentioned earlier in your intro video, incomplete dominance, codominance, multiple alleles, and polygenic inheritance. Those are the four that we're going to focus on now. Actually, we're going to focus on incomplete dominance, multiple alleles, and codominance, and then I will just make mention of what polygenic inheritance is. Therefore, you'll be learning how to make some more Punnett squares over incomplete dominance, multiple alleles, and codominance. Let's start with incomplete dominance. In incomplete dominance, we still have a dominant allele and recessive allele. What you have to remember, though, is that the dominance is incomplete. What does that mean? That means that the recessive trait does not get completely hidden. It tries to express itself. So what we get is a blending of the dominant trait with the recessive trait. So for example, and you can see in my example here, if we mix a red and a white, the blending color of red and white is pink. So this exhibits incomplete dominance. So in this particular problem, we have the F1 generation produced by a cross between white flower and white flower pants. All of the offspring come out pink. Okay. Now, that means that the red allele from the red flowers is passed on, and the white allele from the white flowers is passed on. When those two alleles get together, even though red is considered dominant and white is considered recessive, the resulting phenotype is pink. Okay, this is what we call incomplete dominance. So in the case of incomplete dominance, you will now have three phenotypes instead of two. The only way to have the dominant phenotype is to have two dominant alleles, or two big R's. The only way to have the recessive phenotype, it is to have two little r's, or two recessive alleles. And then the third phenotype is the hybrid or heterozygous genotype of dominant and recessive big R, little r. So again, incomplete dominance occurs when the two alleles blend to produce a third phenotype. So the dominant allele does not completely mask the recessive allele. That's the first thing you need to remember. Second, a heterozygous genotype produces a third phenotype that is somewhere between the other two. In this case, red and white give us pink. Punnett squares work exactly the same as they did before. So in this case, we're going to start with a pink 4 o'clock. A 4 o'clock is a type of flower. It's crossed with another pink. Okay, again, we have our dominant red or recessive white. Since both flowers are pink, the only possible way to be pink is to have a heterozygous genotype. So we're going through, again, just like we did before, the 5 steps. Now we set up our Punnett square. First plant across the top, second plant on the side. We mix our genes or alleles together inside the box. So we have pure dominant, we have heterozygous, pure recessive. So what does that mean? What's the chance of getting red? What's the chances of getting a pink flower? What's the chances of getting a white flower? If we look in there, we see only one that has two big R's. Again, that is the only way to get the dominant trait. So there's a 25% chance. How do we get pink? Pink is the result of a heterozygous genotype. We look in the square. There are two. So that's 50%. How do we get white? White is recessive. The only way to ever get a recessive trait is to have two recessive alleles. So we have two small r's, and that is 25%. So very simple. Do the Punnett square. Again, what you have to remember, if you get a heterozygous genotype, you're going to have a third phenotype. That is incomplete dominance. Codominance. What does that mean? That means that actually we have two different alleles that both want to be dominant. We can see here in the F1 generation produced between a black feathered chicken and a white feathered chicken is we get a black with white speckled feathers. The white wants to be expressed because it is also a dominant allele. So we have two dominant alleles. Okay. A chicken that has this black color white is called an erminate. Okay, that's just its name. Okay, so we can see here, in order to be white, just like we talked about incomplete dominance, you're going to have both big W's. Since white is considered a dominant allele here, it does get a capital letter. The only way to be black is to have two big B's. Again, black is a dominant allele, so it gets capital letter. So we are using two different letters here. Now, the speckled is a mixture of the two dominant alleles, so it gets a heterozygous genotype, big B, big W. So black with white speckles. Okay, so again, we have the third phenotype showing up whenever we mix the alleles. Again, both alleles are dominant. That's a key part here. So we're going to have two capital letters. The heterozygous genotype produces a third form of phenotype. Okay, both versions want to be expressed, so we get a third version. It is not like in incomplete dominance where you blend them together. Okay, these can come up in different ways, but we do get a third version. Okay, just like we had here with the chickens before. Punnett squares work them exactly the same. So we have two erminate chickens. If you remember, those are your black and white chickens. So big B for black, a big W for white. Again, two different capital letters because of codominance. Both chickens are erminate or black with white specks. So we get a big B and a big W. Set up your Punnett square, work it exactly the same. First chicken on top, second chicken on the bottom. We fill in the squares. Be consistent with what letters you put first. And then we simply go through. If we look here, we get two big B's. So we're going to look for black, we're going to look for speckled, and we're going to look for white. So two big B's. Only 1 out of 4, 25%. Two of them with the heterozygous genotype gives me 50%. And finally, the two big W's give me white, 25%. So now we have covered incomplete dominance and codominance. We are now going to move on to what is called multiple alleles. So, what happens in multiple alleles? A single gene can have 3 or more possible different alleles. One we're going to work with today is blood typing. It has 3 different alleles that we will work with. That's probably the simplest one to understand. Okay, so again, we're going to deal with blood type, and we're dealing with 3 or more possible alleles. Because of that, we can get all these different types of blood types. Now, we are not worrying about the rhesus factor. That is the plus and minus. We're simply worrying about A blood, B blood, AB blood, or O blood. Okay? The rhesus factor comes in with a whole different pair of genes. So that would be actually polygenic inheritance when we get to talking about blood type and positive or negative. So we're just going to focus on the blood type right now. Phenotypes include, as I mentioned, A, type A blood, type B blood, type AB blood, or O blood. Again, we're not worrying about the plus or minus. What we have here when we do this particular type of multiple alleles is both complete dominance and codominance working at the same time. That's why I did those two first. Okay, we have a mixture of codominance and complete dominance working at the same time. So how is that possible? It is because there are three different alleles. The allele for type A blood is considered a dominant allele. The allele for type B blood is considered a dominant allele. So we have codominance at work. The allele for type O blood is recessive. Okay. Since we have that, we have complete dominance at work. And we have codominance at work. Now, because of that, we have to letter these a little bit different. We have to show our complete dominance with the capital I's and the little I for O. And then we show our codominance by either putting a superscript A or a superscript B, depending on whether it's an A allele or an AB allele. Okay, once you work one of these, it kind of makes sense. Okay, so we have both complete dominance and codominance at work at the same time. This does result in multiple genotypes and multiple phenotypes. We mentioned earlier there are 4 possible phenotypes. There are more than 4 possible genotypes, so we'll get through all of that here. If you have type A blood, you can be pure for type A blood. That means you have 2 dominant A alleles. Or you can be heterozygous, a dominant A and a recessive O. For B blood, same thing. You can be pure dominant, or you can be heterozygous. The only way to get A blood is through codominance. You must have an allele for A and an allele for B. The other way to get type O blood is to have 2 recessive O alleles. So we have 4 phenotypes, 6 genotypes. Okay, so let's move through this. Punnett squares are worked exactly the same. We go through the same 5 steps. We have a man here heterozygous for type A blood. A woman is heterozygous for type B. We know then if he is heterozygous, heterozygous means 1 dominant, 1 recessive allele. He has a capital I superscript A with a little I. The woman is heterozygous for B, so a dominant B allele with a recessive O allele. And then we set up our Punnett square. We set the man up top. We set the woman on the side. We put them together. You'll see this first one has an A allele and B allele coming together. We have a B allele with an O. We have an A allele with an O. And we have a little I. So what does that mean? What's the chance of getting A blood? Well, this bottom left-hand corner right here is going to give you A blood. What's the chances of getting B blood? What's the chance of the AB blood? What's the chance of getting O? Okay, so we go through our square. The first one there, as I mentioned, bottom left corner is going to be A. We look at the top right corner, that's going to be B. 1 out of 4 is 25%. Top left corner gives me AB blood, 25%, and the bottom right gives me 25% for O. Okay, and that is how you work your multiple alleles. Okay, last thing I want to talk about is something called polygenic traits. Again, this is where you have multiple pairs of genes coming together. Okay, and I'm sorry, I should have said this is controlled by 2 or more gene pairs. Polygenic means many genes. Okay, so anywhere from 2 up to 7, 8, 9 different pairs of genes can affect this. Because of that, we have a wide range of phenotypes. In many cases, what we are dealing with is something called incomplete dominance. Okay, your height is a result of polygenic inheritance. Your skin color is the result of polygenic inheritance. Weight, hair color, all of these require multiple pairs of genes. Okay, Finish the worksheet, get it submitted. Have a great day.